Good afternoon, and welcome to the great aula of Maastricht University. Thank you all for coming for this defense. What you are about to see is an academic dispute, an ancient ritual, but first and foremost, a celebration of science. These little booklets, they seem so small, but willing them into existence takes an awful lot of effort and time and perseverance and ingeniousness and all of those things. And what we're here for ultimately is to celebrate that. So may I please ask you all to turn off your phones, because nothing is more annoying than when you're really in a good flow to be disturbed, and enjoy the show. Hereby I open this academic ceremony in which Ramiro Marroquin Garcia will defend the academic thesis titled Phosphorus-based polymers for drug delivery and sensing applications. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you, Rector, for the introduction. Dear members of the committee, dear colleagues, friends, and family also in Mexico, it's my pleasure to present you with the results that I've been doing in this research over the past almost 5 years. First, I would like to give you an outline of this presentation. I will start by giving a small justification on why phosphorus is so important. Then I will briefly discuss the importance of polymers and monomers. And then I will dive into the specifics of the experimental chapters, which are the drug delivery hydrogels, polymers for silencing detection, and I will finalize this presentation by giving you a summary and conclusions, and also highlighting the potential impact of this research. So let's begin. I would like to draw your attention to the fact that phosphorus plays a major living role for every human being in this planet. All of us have DNA. DNA is a polymer that is composed of nucleotides and an arrangement of sugars. And most importantly, it has this double helix structure. The double helix structure is held together by numerous phosphate bonds, which provide DNA with an outstanding stability. It is this bond that serves as the main focus of this thesis, and its incorporation into functional materials for several applications is the aim of the thesis. So, but what are polymers anyway, and why are so important? Polymers are chain-like materials consisted of various repeating units called monomers. Polymers are everywhere. They facilitate our food supply, our mobility, and also healthcare. And as you can imagine, the monomer selection plays a crucial role on the final properties of the material. In this thesis, I focus on two commercially available phosphorus monomers, the first one being BMEP and the second one BPA. These two molecules have interesting properties. The most important for this thesis is the fact that in the presence of water, they will have a negative charge, as you can see in the image. This charge is so persistent that even after polymerization is there, and gives the final materials outstanding water compatibility, the ability to bind positively charged molecules, and also they are highly resistant to hydrolytic degradation. In this thesis, BMEP was utilized mainly for the production of hydrogels, for drug delivery and sensing, and BPA only for the production of sensors for drug detection. So now I explain you a bit of the experimental chapters. The first one is on drug delivery hydrogels. Hydrogels are a specific type of polymers where the chains are arranged in a mesh-like structure, as you can see there. You're most familiar with hydrogels, I hope, because they're highly commercially available, such as this wound dressing patch that you can put on the skin. In this case, the hydrogel protects the skin, moisturizes, and also has the ability, in some cases, to release pharmaceuticals that can improve the healing process. With this application in mind, the first chapter is focusing on the development of novel hydrogels for wound dressing applications, having a BMEP content more than 70% in weight, whereas the current materials have a BMEP content of just about 30%. That's the research gap we're trying to address. So I'm going to describe you some of the results that I obtained for this research. The first one is that depending on the concentration of BMEP, we could obtain different porosities. As you can see in the screen, we can go from a very close structure on the right to a very open and porous distributed on the left. This has major implications for wound dressings, as the properties are dictated by this, such as the elastic response or the mechanical properties. In this graph, you can see 5 different hydrogels. The one on the top are the stronger, and the one on the bottom are weaker. However, or most importantly, all of the hydrogels synthesized in this have similar mechanical response as the one in skin, which is important if you want to put something on top of your skin for it to heal. They were also highly water absorption. They had a lot of water absorption, and they were able to release and absorb drugs in a very prolonged and pH-dependent manner. The key takeaways of this research is that a library of highly tunable hydrogels for wound dressing applications was achieved. The second part of this thesis focuses on the detection of psilocin. I'm going to explain you the problematic and then dive into the specific way to tackle it. Psilocin is a veterinary sedative. It's a non-regulated substance. That means it can easily be acquired by common people. And in that case, it's used as an adulterant in other drugs. The first one being alcoholic drinks, where the addition of psilocin leads mainly to sexual assault. And the second one being a synthetic opioid called fentanyl. In this case, the mixture of psilocin and fentanyl, it's also called tranq, and it's highly addictive and it can be deadly. This is the reason why it has made it into the news since 2021. As you can see here, it's called the zombie drug, and it eats your skin. As you can see the leg over there, which is not very nice. And this is the reason why the DEA issued a public safety alert since 2021. There are ways to detect psilocin that are commercially available, such as these test strips, which are very similar to a COVID test. However, these are mainly used for forensic purposes. They have poor tolerance to matrix interference, meaning that psilocin has to be in a very pure state in order to be detected. And they also have a poor shelf life. So with those 3 things in mind, polymers or polymeric sensors could be of beneficial, as they are known to be more robust and cost-effective for the detection of small molecules. In this thesis, I focus on what it's called molecularly imprinted polymer, or MIPS-based sensors, and I'm going to explain you what I mean by that. If we consider psilocin to have the shape of this yellow figure over there, if we mix it with monomers in a specific solvent and then polymerize it, we ended up with a solid that has encrusted psilocin in its structure. We can clean the polymer, and then we expose nanocavities that are specific to the molecule. The problem, or the analogy that I can use, is this children's toy. Every figure represents one molecule and only fits in one space. The problem is that polymers alone are not enough for detecting or telling you if you have psilocin in your drink, so you need a readout technology. In this case, an optical approach was undertaken by loading the MIPS with a colorful molecule, such as a dye, and then exposing these materials to psilocin. Psilocin will retake its place, and the dye will be displaced, allowing the visual detection of psilocin by a change in color. With this, the first application was the development of a colorimetric sensor in a portable way for psilocin detection in a gin and tonic sample. Here, I utilize the BMEP hydrogels that I described you in the previous chapter, and they will serve as a trapping mechanism for the dye and also as a reading indication zone. As you can see the components of the sensor here, you can see on your right how the test looks before and after being exposed to psilocin. A change in color from white to blue represents basically that the gin and tonic had psilocin in it. The key takeaways is that this is the first colorimetric sensor for psilocin. It has high tolerance to matrix interference because it works in a complex gin and tonic sample, and it was enabled mainly by the BMEP hydrogels. The last part of this thesis focuses on detecting psilocin, but in a fentanyl mixture. I just explained you the colorimetric approach, where a change in color represents psilocin presence, And now I'm going to explain you a thermal approach. In this case, what you see here is the effect size versus the concentration of xylazine. The effect size is mainly the change in temperature when in solution is present xylazine. If you heat the solution that contains the MIPS, the xylazine will bind to the surface and will block the temperature or the heat flow, and that will lead a change in the temperature. And we compare this to a non-imprinted polymer. That means a polymer that has no cavities for xylazine. And you can see in the graph there that the change in temperature for the NIP, the purple one, is much less than the green one. The colorimetric approach, in this case, was able to distinguish the presence of xylazine in a complex fentanyl mixture that has at least 5 components. We did this because it's a street-like sample, so we made it ourselves in order to test this sensor. So with this, I would like to give you a summary of what I did. In Chapter 2, hydrogels for drug delivery and trapping molecules were developed. These hydrogels were used to develop a portable fast sensor for xylazine in gin and tonic. And the last chapter was a complementary technique using colorimetric and thermal detection of xylazine in a fentanyl mixture. All of these have some limitations and can be improved by the hydrogels need to still be more resistant to breakage. The test for the drink has to work in colorful drinks, of course. And in the last chapter, calibration with a real street sample, it's needed, and also different concentrations of fentanyl. Nonetheless, the impact of this research could have a lot of it in the fentanyl story. As you can see here, consumption of this drug leads to this damage in the skin that can be prevented by using these two polymers in the way of a drug-loaded hydrogel that can aid into the healing process, a fast portable detection for people that don't want to consume it, and also drug checking facilities that can prevent the widespread of this drug. With this, I would like to thank all the people that support me during this journey. Thank you so much to Sensor Engineering, all my family back home. Thank you for your attention and your presence here. And I give back the word to the director.
Thank you so much for this beautiful presentation. Very clear and very good slides. The opposition will now be opened by Professor Dr. Lee Bouwman, full professor of clinical engineering at Maastricht University.
Thank you, Prorector. First of all, I'd like to compliment you on a very impressive thesis that I read with great pleasure. Not only would I like to compliment you, but also, of course, your promotion team. But the time I spend giving compliments is subtracted from the time I'm allowed to have a clear discussion with you, so I'd like to start with a discussion. And I'd like to do that by turning to page 103. And in your tests on page 103, you used interference. And these interference for the test were caffeine, lidocaine, and mannitol. And if you go to the properties of these different molecules, caffeine is very amphibic molecule, so it's both soluble in water and has lipophilic properties. Lidocaine is hydrophilic, or lipophilic, I should say, and often gives cross links and results in a positive test, a false positive test. And then you've got mannitol, that's hydrophilic. But if you develop a drug test, I'm not so interested in false positive tests. I'm quite interested in false negative tests. So how exactly would you tackle that in the future? What should we start developing now in order to get rid of the false negative tests?
Highly esteemed opponent, thank you for the compliments and also for the question. Yeah, so indeed, lidocaine, as you can see in this graph, has some false positive effect here. So you see some color just by the presence of lidocaine in there. So if I understood correctly, the question would be the false negative, so the absence of silicene.
Absolutely.
Right? So yeah, that's also something that I saw during the development of these sensors. The reason why, or the main reason why I attribute this is to the polymer itself. So the polymer will interact mainly with silicene, but it can interact with many things in the solution. Not only lidocaine, not only caffeine, but just ions like sodium or potassium or things that are present in a complex mixture of drugs.
Well, that's exactly my second question, of course, because you used a very specific solution. But how realistically will it react in a complex solution?
Yes, yeah, that's indeed. So the sample that I use for fentanyl has a citrate ion and also sodium chloride present in it. And we saw a huge amount of color being released just by being exposed to those two things. So citrate and sodium chloride. One is the pH. So the polymer is an acidic polymer. So it will have certain response based on the change of pH.
Yeah, so that's work that has to be solved in order to get this up and running.
A buffer solution will be ideal.
Yes.
Keep constant the pH while maintaining all of the samples. However, complex mixtures, street samples,
Yeah, and you referred to that in your
Yes.
presentation, of course, because street mixtures are not clear; they're very complex. Then turning to chapter 3, I'm very interested in what would it take to make this commercially available, and what are the greatest challenges for a dipstick test? Is that the stability, or is it the reproducibility, or the detection level, or can you shed some light on that subject?
Yeah. So I think this is a general consideration for MIP-based technologies. The reproducibility of the polymer process, it's still not quite, it's not robust. So that means inter-batch variation, it's quite large. That's one thing. So you can make a good MIP today and then tomorrow it wouldn't work. I think that's one of the big disadvantages. For this specific dipstick, I think the immobilization of the MIPS in a substrate that allows the displacement in a fast and clear way, that's the second challenge. So how can we ensure that gluing the MIPS into a substrate doesn't displace the dye already before the silicene is there?
Before it touches the gin and tonic.
Exactly, yeah.
Let's say. And you said that the speed, because it takes 4 minutes. And when I'm in a bar, but that's something maybe, that's my negative. I usually take 4 minutes to finish my drink. So is there any process to speeding up that, or is that very difficult technically?
Yeah.
And should I perhaps order 2 drinks and then start drinking 1?
Yeah. I wouldn't recommend to feel dizzy first and then think, something like that. But I would say, so the test itself in the lab, in a vial, takes 4 minutes.
Yes.
A dipstick, 30 seconds.
Ah, right.
That's already
okay.
comparable to...
So okay there.
Yeah, so 30 seconds will be enough, I hope. But if you're very thirsty, probably not.
So then it wouldn't surprise you that I'd like to turn to chapter 2, because I'm a vascular surgeon, and of course I deal with a lot of wounds, and both ischemic wounds, infected wounds, chronic venous wounds. And I was going through the paper, and you tested it specifically on a pH from 5 to 9. Why these pH levels?
As far as I understand, a wound can have this range in pH.
Yeah, but if you get an infected wound, then bacteria can completely get it off the grade. It can go from 1 to 12. Or, basically, this is these are the pHs. But how does that influence then the phosphate group degeneration? How does the pH go into that that solution?
Yeah, that's a very good question. Thank you. So the phosphate bond in these hydrogels was quite stable for over a month in pH 9 conditions. And as I mentioned in the introduction, so DNA has this bond present in it, and DNA rarely loses its structure just by a change in pH. You need some very specific X-ray mutation kind of thing. But so for these hydrogels, I don't foresee a degradation just by the, for example, increase in pH to go to 12, which will be the most attacking conditions. As the polymer itself is acidic, just by being present in water, it will drop the pH on its own.
So it's not the acidicity.
No,
it's the other.
It's the basicity. Yeah,
absolutely.
Part.
So I'll try to get you out of your comfort zone a little bit, because you're a very good scientist. And if I were to ask you, how exactly would I want to study your hydrogels in a clinical setting? How would you put up a study? How would you make me convince that my off-the-shelf hydrogen, which I use, hydrogel, sorry, which I use quite often, that yours is better? How would you put up a study like that? So I know that you're in the laboratory setting, and I'm asking you now to put a study in a clinical setting.
Yeah. Well, so first I will try to get some ethical clearance for the experiment. That will be my first. That will be my first step.
Very wise.
And then I will run controls with a hydrogel that is the standard in a clinical setting, for example, against my hydrogel, and also a control without any hydrogel supply. Maybe just if we're talking about the drug-releasing hydrogel. Yeah. Then just the drug intravenous as a control or as a...
And you'd probably want to do that in a double-blind setting, so not telling me what you're using.
What I'm using, yeah.
Yes, very nice. Okay, thank you very much for your answers, and I'd like to give the word back to the prorector.
Thank you so much.
Thank you, Professor Baumann. The opposition will be continued by Professor Dr. Lorenzo Moroni, Director of the Merlin Institute and Professor of Biofabrication for Regenerative Medicine at Maastricht University.
Thank you, Mr. Prorector. Dear candidate, also my congratulations for a very nice piece of work to you as well as to your supervisory team. I also read it with great pleasure, and also personally it's very nice to see you here since we know each other for a little while. I'd like actually to follow up a little bit on the previous opponent, but then I'd like to also make it a little bit more entertaining and ask one of your paranymphs to read proposition number 7.
Most reported MIPS recipes work excellently on paper.
All right, thank you. So without asking Casper, which is the follow-up proposition, can I ask you actually to elaborate on the non-paper part of the proposition? So what are the challenges that you face? You alluded a little bit also in your previous replies, but can you go a little bit more into details? What are the challenges in making this technology more reproducible, more across different batches, and so that it works both on paper and in the lab?
Highly esteemed opponent, thank you so much for your words and for the presentation, for the question, sorry. Yes, so most of the MIPS recipes that are in papers or published already have a difficulty to be reproduced. That's my experience. That's why I wrote that. And I think also some people here may attest to that. Mainly because the procedure is not clear. And also the way that the MIPS are synthesized is a bit just simple. It's a vial that you mix, and at the end you get a solid polymer. You grind it and you test it, and if it works, fine. There's no way to characterize it chemically, let's say NMR, or it's very difficult as it's a highly cross-link.
Because correct me if I'm wrong, I mean, if I remember correctly from the theory, also I did also a little bit of work on soft MIPS. Actually, you take your material of choice that should not be polymerized yet, you mix it with your molecule that you would like to then sense. You polymerize the mixture, and then in principle you should remove the chemical that you have added to your pre-polymer, and that in principle, I'm sorry, I'm not going to be simplistic, but also to challenge you a little bit in the discussion. And then, that's it, right? So then you remove your chemical, and then you should have these kind of cavities that are both geometrically as well as chemically recognizing the same species as the same species comes back into or comes back in presence to your sensor. Am I correct?
That's correct, yeah.
So in that mechanism, what would be the most challenging part? Is it the chemical recoupling, or is it the geometrical feature that is not really quite maintained, let's say, after the molecule is being extracted?
Yeah.
Or another variable?
Yeah, I think there are both. So the shape cavity, there has been no way to quantify that you have a cavity that is the size of the molecule itself. So you cannot see that in microscopy or any technique. So you rely on, well, yes, it's a jump of faith that is there.
Yeah.
And then the second, is it's mainly hydrogen bonding. So hydrogen bonding can vary dramatically depending on just...
Clear, yeah, but I think this is the answer that also makes me more convinced to understand better also where are the challenges, right? Because indeed, it's being very dynamic.
Yeah,
it's also very difficult to then always at every single batch reassess it, also with analytical techniques, as you say, right? So because I still got excited reading your thesis, and then if I would immediately imagine, oh wow, could you maybe even make a MIP that has multiple sites of recognition? So could we go there? Do you see that as a future research venue of having a sort of multiple species in one sensor kind of device?
Yeah, definitely. I think that's already been done. Yeah, not only for similar molecules, but very distinct ones.
Okay.
Yeah. Chemically distinct.
Okay, and then if we would want to go out of the lab into an industrial setting, what would be your recommendation to a company actually to try to make it as more reproducible as possible in such a setting?
Yeah, so I think that goes back to the previous answer. The polymerization technique that you use for the MIP production is, in my opinion, one of the most important parts. Depending on the application that you want, for example, if you want a dipstick material, so probably the approach that I took, where you have a solid powder, and then you need to somehow mix and spread, and that wouldn't be ideal. But there are techniques where you can electropolymerize, and then it will be bound to the surface. And you can easily do that in a more repetitive, in theory, way.
Great, thank you. Now, let me maybe ask you a little bit a different question. You may expect that from me, but I think across the thesis, I have seen plenty of different graphs, some differences that are outstanding, and clearly I would have expected some sort of signals of statistical analysis, but that is somewhat something that I have missed in the thesis. Could you maybe comment on that?
Yes, Lorenzo. Yeah, that's indeed. I don't have ways to defend on that. It's not there, mainly because of the lack of need for it in the papers that we publish.
Yeah,
but it is
necessary. By background, so I understand where you come from. But you do realize that maybe thinking about it, maybe a second or third time, it might be nice to maybe just put the Okay, clear. So if I look, for example, at figure 2.8 in chapter 2, this is page 51, I guess that you would probably have statistical differences when you change your comonomer from S1 to S2, but probably S3, S4 are not really having such a clear-cut difference across the pHs, sorry, I should mention. And then again, you see somewhat a more remarkable difference in S5. Am I correct?
Yeah, that's correct.
And if again you look at this graph as actually actually this is quite a nice readout, right? So I mean, essentially you can, in this case with this APTAC hydrogels, release your drug of interest for about 40-50% of its content. What happens to the remaining 50-60%? Do you expect it to be released at a given time, or will it be always entrapped in the matrix? And this is in a way the efficiency of your system.
Yeah. Well, so I didn't test beyond that point, but I can comment on what I observed before. So I think the drug will be retained in the hydrogel, and if there will be a release, will be very small. As you can see, in 72 hours, the graph reaches, most of the graphs reach
A plateau.
complete, like to a plateau more or less. So I don't think it's gonna be released.
Yeah. I must admit myself, I haven't looked too much into details on the chemistry of the APTAC, but you don't see that as being degraded in the long, after a year. Okay, now you have conducted the experiment for a week or 8 days, I say.
Yeah.
If I make the math correct.
Yeah, it's 7 days, yes.
Yeah.
Well, so it's an amide polymer.
No.
So it won't be degraded hydrolytically, and also not the phosphorus part. So I would say it will be stable. The problem I foresee is that the longer you keep these hydrogels in water, they will just start to crumble.
Yeah.
And tiny particles will be appearing, and then maybe more drug will be released afterwards. After it's completely.
You may have actually some release,
breakage.
any kind of terraces level.
Yeah.
Right. Okay. Thank you. I give back the word to the prorector.
Thank you.
Thank you, Professor Marroquin. The opposition will now be continued by Dr. Ali de Boer, Associate Professor of Nutrition, Food and Information.
Thank you. Dear candidate, first of all, I would also, of course, like to congratulate you with your work and also extend my congratulations to your supervisory team. As a nutritional scientist with an interest in how we use science in regulatory procedures, it's also with that perspective that I read your thesis. And it's really interesting to see how you work on rather fundamental applications and study how you can make sensors work in a real-life setting. And it's that translation that I'm really interested to have a discussion with you today. Because if we look at what type of evidence would be needed about your sensors and about your applications to actually make them work in real life, and I think the two other opponents already discussed some of these aspects with you as well, we probably need a bit more details, right? And we need to identify a few more strengths and potentially work on some weaknesses of these sensors. So I would first like to ask you a bit more about the proof of concept versus real-world evidence that you present. If you look at the different applications that you worked on, and in a more general sense in your thesis, the standard of evidence, like I said, in the regulatory context and in the decision-making context is often a bit higher than merely showing that something works in a lab. How do you look at that research gap? What kind of work would be needed in testing your applications to move towards that more acceptance phase of the technologies?
Highly esteemed opponent, thank you for the question. In that regard, I think, not to repeat myself in the previous ones, I think the polymer itself has to be robust enough that can be utilized widely in the field. The sensor has to be designed in a way that is the same weight of polymer goes into, say, the same dipstick. And in that way, by establishing protocols for the MIP synthesis and the device fabrication, we could go and test in a real setting. So it's mainly focusing on how robust is each step of the process to give some confidence that it will work in the field.
To make sure it's indeed reproducible in all these different settings. Okay. I would like to move with you to a more specific part of your thesis, chapter 3, building on this first insight. And I would like to invite one of your paronyms to read proposition 3.
an extremely low limit of detection is not always the most important characteristic in a sensor.
So in this proposition and also in the chapter, you state that an extremely low limit of detection is not always the most important characteristic, but you now also already elaborated that reproducibility is really important. How did you decide on the characteristics that you worked on in chapter 3, the performance characteristics? What kind of characteristics were most important to you in your first testing?
Yep. Yes, that's a very good question. So in chapter number 3, the focus was basically to understand what type of test we needed. So what concentrations are the most relevant. That's why I wrote that the extreme low limit of detection is not the most important, because sometimes the limit of detection that you require or that it's present in the real setting is much higher than what you would aim for. So if I aim for a nanomolar range detection and my real sample spikes at micromolar or millimolar, I do think as a scientist we should strive for being the most, how do you say it, detect the most, but sometimes that can, again, go back into how reproducible and how applicable will be if I try to make it as perfect as I can. So sometimes good enough is enough, I would say.
Okay.
Yeah.
Because especially in a regulatory context, that's quite an important point, right? Because spiking a bit is also completely illegal, right?
Yes.
So that's an interesting point. When is good enough actually good enough? How should we come to a conclusion on that?
Yeah, so indeed, in that case, I will retract what I just said, and I would say we should strive for detecting the tiniest bit of xylazine present in the drinks just to avoid human consumption as the main goal, I would say.
But at the same time, that makes me wonder immediately as well, also based on your discussion with Professor Baumann, to what practical application are we then working towards? Do you imagine that we go to a club carrying a MIP dipstick and indeed testing ourselves? What is the ultimate aim with this development, you would say?
I would say the aim will be that to provide like a technology that is portable, cheap, and fast, so that anyone could do it by themselves. And in that way, well, get the security that what you're drinking is safe.
Would you actually expect that this increases security of people going clubbing?
Oh, that's a good question. No, I think that will be suspicion for everyone to be like maybe just have that on your pocket.
Yeah.
Knowing that you may need it, it's, yeah.
It'd be a bit scary, right?
Might be, yeah.
But it's an interesting thought, right? It made me especially wonder indeed, for what purpose are you developing this? And it might need different approaches, right? You might need different strategies also to come to a conclusion whether it's been developed good enough. But I think that became very clear for me in the discussion. I was wondering about something building onto this. You now tested the gin and tonic example, right?
Yes.
How did you decide to start testing in gin and tonic?
Well, so I tested other drinks which are not present in the thesis. However, as you can imagine, most of the drinks outside have some color. Beer has color, like wine has a lot of color. So in this case, the one that provided the best result for like being complex enough as gin is, I think it has like 1,000 active compounds or something. So, and displaying no color, that was the main reason. Right. Because the color change, it is visible, but if you have a certain interference with color, then you will lose it. Or at least by eye, it wouldn't be so detectable. Gin and tonic was clear, so we used that.
Yeah. Because it's especially that you, if you look at the food and beverage market, very often they are more complex than gin and tonic is, right? So how do we move from a sensor that can purely work in a very clear solution towards a portable device that actually can work in these more complex matrices, which foods often are?
Yeah. Yeah, that's a challenge for these type of technologies, because what I just said, right? So the selectivity or the response will be given even if xylazine, for example, is not present. So some sugars will have this effect of releasing the dye, and then you will have the false positive. However, I do think that here is more like the engineering part of the device. So having several steps where you filtrate certain compounds, certain colorful molecules, if that's even possible, then that will be a way to go. So xylazine being in high concentrations, then you will probably absorb some of it in the filters, but it will pass down to the actual sensor.
Yeah.
So that would be my answer to that. So,
And you mentioned if that's even possible. Do you think it's possible?
I think it is possible, but again, it depends on what type of beverage are we talking about. Yeah. So beer, for example, I don't see beer as a good candidate just because it has so many compounds in it.
Yeah.
And also a very strong color.
So we might end up working with a dipstick with multiple MIPS to actually make use of this.
Yeah.
Okay, thank you very much. I give the word back to the program.
Thank you.
Thank you, Dr. de Boer. The opposition will now be continued by Professor Lilian Lacerda Bueno from the Laboratory of Immunobiology and Parasite Control from the Federal University of Minas Gerais.
Great, thank you. First of all, I'd like to congratulate you, Ramiro, the candidate, and the promoters for this nice work that you have done. And it's really good to be a part of the jury. Thank you. And regarding to the chapter 2, I ask you a question. Could you explain how increase the BMP contents influence the balance between mechanical stability, swelling behavior, and drug release kinetic, and how you determine the optimal composition of drug delivery application?
Highly esteemed opponent, thank you for the question. Yes, so in this work, I would say, the balance between the mechanical properties, drug delivery, and the other swelling properties.
Yes. The swelling behavior.
Are, in my opinion, the best for the pure BMEP hydrogel. And that is mainly because of the chemistry of the reaction, so the polymerization.
Okay.
So having a material that has like a mesh structure, which would be the case for the BMEP hydrogel, pure BMEP 100%, will have a more robust response in terms of mechanical properties, probably more brittle, I would say.
Okay.
And the distribution of the pores will be probably more consistent through the sample.
Okay.
By adding the other monomers, which are charged, one was negative, the other one was positive, then it was more difficult to assess the hydrolytic stability, the swelling, and the mechanical properties. What we saw is that just adding a comonomer will drop dramatically the mechanical properties.
Okay.
So in that regard, I would say in this chapter, BMEP 100% sample will be the best candidate for a balance between hydrolytic stability, swelling, mechanical properties, and drug release.
Perfect. Now we can talk about, actually in the chapter 2 yet, your system, which parameter do you think the most critical to turn in order to precise control the release of profile and therapeutic molecules?
Oh, that's... If I understood correctly, the question was, could you
what I mean ask you, because you have to control the drug release,
yes,
is strongly influenced by the hydrogel structure, the structure.
Yes.
Your system, which parameter do you think is the most critical one to control the release, to precise control the release of the therapeutic molecules?
Yeah. So in that regard, I think for these specific examples, the pH would be the most important one. So having a very basic pH or a acidic pH will, as I show in the graphs, will have a different profile. I would say if the pH remains within those limits, then we can average the tree performance, and then it will be stable. Yeah.
Yeah, okay. Agree with you. And I think that's the most important one, okay? If you had 3 more years to continue this project, what would be the next step?
Oh, yes. The chapter 2 or the whole thesis?
The whole thesis, please.
Okay. Yes. So I was already starting to work a bit on what would be the follow-up before I finished. So to tackle the problem with like colorful drinks, together with a colleague, we were developing fluorescent-based displacement,
Okay.
which will give a fluorescent response using a laser or a portable light, and it will be regardless of the color of the matrix. So it doesn't necessarily have to be a blue shift, but you will see the color once you shine light on it, and you will know depending on that. So that was one of them. And for the hydrogels itself, I was thinking about modifying the recipes for making the stability in a long term, so over several months, much more robust, so that they don't crumble after 30 days being in water. Just moving towards a material that is mechanically robust, that can release drugs over more periods of time. For example, I don't know, maybe use them not for just drugs, but for vaccines or something that can be released more. Yeah.
Okay, that's it. Thank you very much.
Thank you, Lily.
I can go back to the projector.
Thank you, Professor Lacerda Bueno, and thank you also for coming all this way, for being a part of this defense. We really appreciate it.
Thank you.
The opposition will now be continued by Professor Dr. Elisabeth Rosado Balmajora, Professor for Experimental Orthopedics at the Universiteitsklinikum in Aachen.
Thank you, Pro-Rector. Dear candidate, I would like to start by also congratulating you in your thesis and your supervision committee. I'm very impressed with the translational aspect of your thesis. Your applications are clearly an impact to society, and you have, on top of that, published very well your chapters, and you have even publications in collaboration with other institutes. I'm really congratulating on that. I have a curiosity before we dive into the questions. Did you and your teammates make it to drink all that gin and tonic, or was all spiked in? I hope you enjoy a little bit of the drink besides, I guess so.
Yes.
Very good, very good. Then I would like to ask one of your paranymphs to read proposition number 2.
In a hydrogel, a 1,000-fold increase in the mechanical properties can still provide insufficient stability.
Exactly. And if I follow up from our discussions before, indeed I agree with your proposition. However, we keep using hydrogels as one very advantageous type of biomaterials in biomedical application. And this is because the bioproperties are best among many other polymers in the material field. Do you have any suggestion how can we indeed combine best mechanics and best biology in a hydrogel from the material point of view?
Highly esteemed opponent, thank you for the question. Yes, so I think the trade-off between having a material that can absorb water and have good mechanical properties is challenging, because if we move towards the mechanical properties, then we compromise the water absorption or the drug release or just, I guess, cell interaction. So what I would suggest, or what I is to still achieve high or at least relevant mechanical properties for the application, for example, cartilage or bone, so it has to be at least aimed at that specific. And then the use of additive molecules or functional loadings in order to compensate for the decrease in swellability or water absorption, such as, I don't know, proteins or, yes, that would be my suggestion.
I fully agree with you. I think composite materials may be one avenue to improve those mechanical properties while still preserving the bio properties of the hydrogel. I would like to focus in your chapter 2 and a little bit of the biology and the application of your material for wound dressing, because I found this chapter really particularly exciting. You report the preparation of your hydrogel using synthetic. It's basically a synthetic type of hydrogel, right?
Yes.
We have a lot of natural origin hydrogels, particularly attractive chitosan, collagen, and so on. Do you think your materials are advantageous compared to natural origin gels?
Yes, I do think so. So the hydrolytic stability of the mentioned biopolymers is somehow not optimal depending on the conditions. For example, hydrolysis of chitosan, collagen is more difficult as far as I understand. It's more robust. But so I would say the polymers that I synthesize lie in between those of the biopolymers. With the facility or the ability to be synthesized much more with different architectures and different co-monomer compositions, tuning properties, which biopolymers sometimes it's more challenging. So I would say, yes, they can compete with biopolymers.
I agree. So basically, manufacturing is better and perhaps batch-to-batch reproducibility will indeed win. Do you see any limitation, however, on your synthetic hydrogels when you think about chitosan or collagen?
Yes, if the application requires degradation or absorption by the body to lead into a functional tissue, then probably these materials won't be ideal. Degradation, even if it happens, will release some acidic components, which won't be very good inside the body, whereas the other components just release some sugars and then
Exactly.
Yeah.
So there we go into my next question, and we touch toxicity and biocompatibility a little bit, and that link up with the leachable materials on the degradation of the polymer, right? You selected bovine fibroblast in this chapter to do your text. Very good. Thinking about translation and thinking about moving your system quickly into human applications, do you think that the bovine fibroblasts were the best cellular system to test your material?
No, I would prefer to have it in a human cell line. But there were limitations regarding the access to these cell lines.
That is clear, but I'm very happy to the answer. That is what I am looking now, not to redefine your experiments because that makes no sense. But indeed, having a human system will indeed give a little bit more of translation to the material. Another point is we have ISO standard norms, and there will be indeed define perhaps which cellular system will be indeed needed for those regulatory processes that we stressed before. Now, one thing that we almost always forget when we do materials for biomedical application is the clinician, right? Which is actually the final part of the chain that actually is going to decide a lot whether our device it will make it or not, basically. When you think about your wound dressing and you consider the products that are already available, do you have an idea how you will design this to be the final boxed product?
Yes, so at the beginning of the thesis, I also aimed to have a product. I wanted my thesis to have specific or small products out of the chapters. However, looking at what it's out there already, for example, addition or a layer that is adhesive to be stick into the skin was one of the main drawbacks. I thought I had to include it in the hydrogel, and that made things a bit more complicated for the properties. So I would say it cannot be a one material job. So it has to be composed of several functional layers. And in that regard, I think that would be my approach for these type of hydrogels. As I said, the materials alone soak in water for a long time. They just crumble apart. So having them in a protective mesh that ensures that even after breakage, they can still hold their structure will be the first step. Then the adhesive layer, and then that will be my suggestion. But I think talking to the end user will be my first. Now that I think it after 5 years will be my first.
Well, that is a very, very good and very highly desired thing in the scientific community. We very often don't do that. We are learning that is very important. And it's good to see that in such a young scientist already, that's thought. What about lyophilization? Could we just make it dry?
Mm-hmm. Yeah, the hydrogels were made dry at the beginning because we use organic solvent for the synthesis. So we had to leach this solvent, DMSO, that is also not very nice for a cell. And after being cleaned, then we freeze dry them. And that's how I started the experiments. So freeze drying was part of the manufacturing.
That might prevent the crumbling.
In water, yes. Yeah.
I see. And still in chapter 2, when we see your enzymatic degradation study, which I like very much because I did a bunch of that myself in my PhD thesis. You selected lysozyme and 1 milligram per milliliter. Why lysozyme and why that concentration?
Yes. Well, lysozyme and the concentration were based in a literature paper that was testing similar hydrogels. However, I know that this enzyme does not break the phosphate bond. It's an esterase, as far as I know. So it didn't degrade the material because it was not aimed for the specific bond. Nonetheless, it's present in the literature. So I went for what it was like the testing standard for these type of hydrogels.
Very good. So if we connect that answer to the question that was asked before, where you define some experiments that you will do in the next 3 years, where biology was barely there, a bit disappointing. But if we redefine that, I guess we will put enzymes now, perhaps another enzyme, perhaps a cocktail of enzymes.
Yes, that's indeed a good suggestion, and I will definitely. I thought about the phosphatase enzyme before, so yes, that's it. Which I'm not sure if the body has phosphatases per se, but I guess so. No, yes, so that would be the case.
Very good. So moving on. We talk a little bit about the human cells, and I was very happy to see that you were considering we could have indeed some more translational biological system to test your hydrogel. If we don't talk about cytotoxicity, but we go a step forward and talk about functionality. Do you have an idea of an assay or a model that could predict how good is your material in wound dressing?
I would say, I don't know if it's do you mean a standard test or a test on its own?
I mean, your ideas.
Yeah.
Test on his own.
Yeah, so I think we could incorporate these hydrogels into lab-on-a-chip models where we can have not only the cells, but already the tissue that has been assembled previously, and then play with conditions such as the pH or the effect of the complex arrangement of extracellular matrix and collagenases and all these things reforming and being more dynamic than just the cells alone for a short time.
That's a great answer, because thereby you could combine the bacteria and the different pH perhaps and have a more complex cellular system that give you the layers of the skin and how the phases go on. Thank you very much for your answers. I give the word back to the project.
Ramiro Marroquin Garcia, the time appointed for defending your thesis has now passed. The degree committee present today will now withdraw to discuss the quality of your thesis and your defense. And so I request that you and your company await the results of our deliberation and our return in this room. Ramiro Marroquin Garcia. The degree committee here present has assessed the quality of your thesis and your defense today. In view of its positive verdict, and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Dr. van Grinsven is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And therefore I now invite your supervisor to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Ramiro Marroquin Garcia, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Dear Dr. Marroquin Garcia, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. A doctorate is forever, and while you have undoubtedly shown that you've deserved it, its true value lies in everything that you will do with it. I'm certain that you will do wonderful things with it, and we will follow your career with great interest. Congratulations.
Thank you so much.
I hereby close this academic ceremony. Mistake. But I now invite your supervisor to say some words of praise. Ooh, first.
Yeah.
Dear Ramiro, what a ride it has been for you over the past 10 years. You came here from Mexico with your wife, Ro. You followed the master's program in bio-based materials, and after that, you landed a PhD position, but not immediately with us. Early in your career, you had to learn that not everything goes according to plan. At one point, you had to make a choice. Stay in your old lane or change to a new one. Today, I have the opportunity to thank you for the choice you made. Roughly 4.5 years ago, you came to the sensor engineering department and restarted your academic journey. It is fair to say that this journey was not without obstacles. You started with the creation of hydrogels, and due to a variety of circumstances and opportunities, you eventually turned to the dark side. You entered the world of sensing, and you did it with a vengeance. But this would be a rather boring speech if I simply listed your accomplishments. You already did that during the past hour, and instead I would like to talk a little bit about the fight you fought and won. There were times you had to go deep. Too many times were deviations from the plan, and that took its toll. However, some of my fondest memories are the walks and talks we had during those moments. We both tried to give each other perspective. You live by the credo, you are what you do. And I think that is where we connected. You explained to me what this sentence meant for you, and I explained what it meant for me. And from there, we moved forward. But I was in the lucky position of having a few aces up my sleeve to convince you that you are what you do includes more than research. During your time at the sensor engineering department, you became a dad. Twice. On September 20th, 2023, I stood in this very same place and had the privilege of giving the laudatio for your wife. During her defense and during the laudatio, I could hear you and the apple of your eye, Paula, up on the balcony. And during the final stage of your own journey, you became a father for the second time. You and Ro welcomed Emmanuel. Twice I saw how you became a dad, supported your wife, dealt with sleep deprivation, and throughout all of that, you still did an amazing job. Towards the final part of your journey, I noticed a slight change. I saw a slightly softer Ramiro. Research still played a massive role in your life, and given the accomplishments, nobody can deny that. But also our exchanges changed a bit. They were no longer only about research, they also included the kind of well done you stickers that you give to your daughter when you want them to do what you want. Your perspective changed a bit. My Ramiro, you are what you do, and you did great. Don't be too hard on yourself and others. And a small PS, the swimming lesson for Paula was my pleasure. Congratulations.
Thanks.
We wouldn't want to have denied you that. My apologies once again. But now, I close this academic ceremony officially. And, well,